Thank you. It's a pleasure. Let's see. Thanks. This consortium that I'm going to talk about this morning is very much focused on moving from the genomic discoveries to understanding the cellular mechanisms and processes impacted by that genetic variation, and then onto the disease mechanisms. It's a very new consortium with the first meetings being held at Oxford in this December, and with lots of great discussion. The idea is to really accelerate our progress in moving from the genetic discoveries to understanding the driving molecular processes and functions, and then onto the physiological mechanisms for the genetic diseases. And we are focused on common diseases where we have this challenge of many discoveries having been generated over the past 15 years or so, 15 years, I guess, but not yet enough understanding of the driving mechanisms for what is largely non-coding common variation underlying these discoveries. But even to get here, if you'll recall, has just required a ton of infrastructure building, collaborative efforts across the world in generating maps of genetic variation, in generating relatively inexpensive ways of interrogating the genome at scale. And yes, now we finally have not just discoveries, but some insights into mechanisms and pathways of disease that we would lack without the genetic understanding the genetic basis of some of the discoveries. And of course, that's the goal, to take these common diseases, the things that really put people into hospital beds, that send people for medical care, and get an understanding of the biological mechanisms driving these kinds of phenotypes. So the big challenges are understanding how these non-coding variants map to functional units, how those functional units are driving the observed disease processes. And so we go from samples to variants to genes and other functional units. We need to understand cell types and the molecular processes linking the genetic variation to these cellular processes. And then disease-specific mechanisms. And this will take really different kinds of studies. So the rationale, the driving rationale that brought many of us together around these ideas were the big projects like GTEx and the Human Cell Atlas that have been so instrumental in helping us get some understanding of the variation that has been discovered to date in the realm of common disease. And we thought that by having so much more data being generated in more cell types, not just transcriptomics, but translational efficiency, more assay of the proteome, understanding how proteins are modified, the genetic contributions to those protein modifications, protein turnover, understanding those processes at scale, but then being able to apply that broadly across biobanks and cohort studies that have medical phenome at scale, or broadly collected phenome. That's going to give us insights into the pleiotropic nature of the variation. It's striking how nobody bats an eyelash at the pleiotropy, the very massive pleiotropy that we see from Mendelian disease genes, because we understand often why it is that we see many different organ systems affected by mutations in a single gene. We have understood how the violation, the mutations of those genes alters cellular physiology in whatever organ that gene's expressed in to lead to what we would otherwise call pleiotropic effects. But when we see it in the context of common disease variation, it's initially distressing, but I think an opportunity to create much larger targets for discovery. We also see the value of including the GWAS meta-analysis cohorts, where we have much larger numbers of subjects with particular diseases, but then not all of the disease and phenotypes that are collected in the biobanks and cohort studies. And so by interacting, creating an environment where we have the data at scale for the molecular and cellular function information that can be cross-linked to the genomic information and phenome at scale, as well as the GWAS meta-cohorts, we would hope to see a faster progress to understanding disease mechanisms. And this is really the heart of what we'd be trying to create in terms of all of these data around the molecular and cellular processes. I think it's that this part of it is a quite ambitious goal, but I think nothing less will be required to really get to an understanding of how common variation really drives disease process. So as I said, we had an initial meeting in Oxford in December, and the consensus to continue building this consortium, and the meeting report can be seen here. We have several working groups now, so a human variation working group, where the idea is to really go from genome to phenome in case control meta-analysis data, cohort studies, biobanks with electronic health records, and other broad phenome. A systematic catalog of variation to function, so trying to connect DNA variants to functional units through the operating mechanisms, and then getting to disease-specific mechanisms given that information, so connecting genes to cellular pathways and general physiological mechanisms. We are also cognizant of the need to create this in a platform that really brings young people, the next generation as it were, together to do the computes over these data. It's a different world out there in terms of data science and the way research is being done in computational biology, and the opportunity to create data platforms that will support this kind of research, we think certainly is within reach now and is what we have to do going forward. And of course, the data sharing and ethics, which has been discussed considerably already today. So we're trying to engage the global scientific community. I really just come almost directly from Taipei, for example, and so we've got 30 individuals from around the world engaged in a proto-organizing committee to build this consortium in a global context. Ongoing discussions with complementary consortia, really no need to rebuild any wheels, so we're trying to maximize synergy and minimize overlap. We have our next planning meeting in Helsinki next month, and then in September, a public meeting in Washington. So that's sort of the rationale for the existence of this consortium, and I'm happy to take questions about any aspects of this that people are interested in hearing more about.
Rory Collins, Oxford. Nancy, from a cohort perspective, what are your thoughts in terms of a scalable approach to getting clues about the link between the genetic hits and the disease, where you've got thousands and thousands of hits? Do you think that the next stage might be to do some kind of agnostic omic assays across cohorts that might identify some of those clues, you know, in the way that you might pick up LDL being the link between genetics and coronary disease?
So I think, so were it possible, from existing human cohorts, to be able to do the assays in as many cell types and tissues that they probably need to be done in, that would be a fantastic idea. I think it's critical to go deep, as say with the GWAS meta-analyses, where we can in cohorts, especially those that have a lot of phenotypic depth to them. But I think we cannot escape really doing as many different kinds of cell types as possible. One of the things that we're really seeing in GTEx is the shared genetic architecture that we see across tissues is often a consequence of cells that share developmental ontologies and genetic architecture around all the processes that are happening in those cells that end up in quite different tissues. And it's really that sort of thing that drives, what people will call horizontal pleiotropy of the same genetic variation affecting translational efficiency in gene activities or transcriptomics or other things. And so I think doing assays in many different cell types is something that we can't necessarily avoid, at least at the tops of these cellular ontologies. And unfortunately, it's very hard to collect all of those cell types from living individuals, from existing cohorts, for example. I think we have to do both because it's tremendously important to see some of these in the context of different disease phenotypes, but also to have the breadth of cellular mechanisms really done in as many cell types as possible.
Yeah, it's just given, Jeff Ginsburg, just given what you just said, have you discussed the role of iPS cell collections within the context of cohorts? Have you?
Yeah, no, so I think there's a real desire to see how well we can do at being able to create IPS in a lot of different contexts. I mean, by having large biobanks and cohort studies, the opportunity to get into far tails of polygenic liability distributions, to be able to create IPS knowing the backgrounds in different contexts, so we can get mid-ranges and one standard deviations from the mean and far out into the tails, I think creates tremendous opportunities to better understand also the function of rare variants that can be CRISPRed in the context of known polygenic liability chosen, as it were, from different parts of a distribution. So I think, yeah, being able to query all of the contributors with respect to how likely it is that we can establish IPS in, if not specific individuals, at least individuals from chosen ranges of different kinds of distributions, I think is really important and another key deliverable for something like this.
Could I just ask in the room if anybody is routinely or maybe episodically collecting iPS cells in cohorts right now?
Yeah, no, and I think we're trying to set up the mechanism broadly within BioView.